Welcome everyone to the RNA Innovation Seminar Series from the University of Michigan. Today we're going to feature two rising scholars. The first speaker is Yousuf Khan. He's a graduate student from in the Molecular and Cellular Physiology program at Stanford Medical School or School of Medicine. Yousuf got his undergraduate degree from University of Maryland. He got a lot of awards and fellowships, and he also is a decent tennis player. So he played for their club team at Maryland. He then did a master's program at the University of Cambridge. And that's where he started working on ribosomal frameshifting that we're probably going to hear more about with Andrew Firth at University of Cambridge. But today he's going to talk to us. He's actually coming from Sweden. He's visiting there. And I hope everything works fine over there in Sweden, all the way to the seminar. And he's going to talk to us today about CCR5 as a model to examine reporter assays in evaluating translational phenomena. Welcome, Yousuf.
Hi, thanks for having me, and thank you for that wonderful introduction. So let me just share my screen and we can get right into it. So, like we said in the intro, today my talk will be titled CCR5 as a model to examine reporter assays in evaluating translational phenomena. And I think this talk is more of a, I guess, a cautionary tale and kind of a study in terms of, really controlling for, the system you're using and being very careful in interpreting your results. Here I like to call them my sponsorships, whatever, just put them up at the beginning. And this is a view from Churchill College at the University of Cambridge, where I did the majority of this work, which was very beautiful. And I met a lot of really wonderful people. And I'll just get right into it. So first of all, I just want to talk about gene number versus complexity. And please let me use the word complexity in a very, very loose manner. So on the left hand here, we have a younger version of me, an embarrassing photo with a bowl cut. And on the right, we have a banana. Now, using the very loose term of complexity, I would hope you guys would think that maybe the person on the left hand is a bit more complex than the banana on the right hand side. But if we look at the number of genes in humans, depending on who you source, anywhere between 19 to 22,000 protein coding genes, depending on your definition of a gene. And for this species of banana or other species of bananas, you're looking at around 36,000 protein coding genes. So, what gives, what's the difference here? And so if we go back to our high school biology textbooks, we look at the central dogma, which is when we have our DNA, we transcribe it into mRNA. This mRNA is then translated into a protein sequence. Now, we have this thing called alternative splicing, which allows us to make many different transcripts, and which can then be translated into different unique protein products. So transcriptional diversity, really great, really well studied. And if we go to the NCBI RefSeq today, there are over 100,000 confirmed reference sequences for mRNA transcripts available for Homo sapiens. So lots of different transcripts, lots of different proteins, and lots of different complexity, and I use complexity in quotes, that we can generate. But what I want to talk about today is something called translational diversity. So here's just a really, really representative sample, the whole host of different translational phenomena that contribute to translational diversity. In addition to post-translational modifications, there are these different phenomena that can create completely different types of protein sequences. So if you follow me from the top right, we have ribosomal frameshifting, which I'll talk more about later in this talk, where during translation, the ribosome will shift back typically one nucleotide, though there are other flavors, but -1 ribosomal frameshifting is by far the most common and well-studied phenomenon, which the ribosome will slip back one nucleotide and continue translation in the -1 frame to create a 0 and -1 frame fused peptide. There's also stop codon read-through, in which the stop codon is read through. So this typically happens with UAG, with a specific, RNA context that allows the ribosome to incorporate an amino acid, keep reading on past the stop codon, and make a C-terminally extended protein. There's also translational bypassing, where the ribosome during translation will shunt over several nucleotides and continue translation and completely bypass parts of the coding sequence. We have internal ribosomal entry sites. And so here is the secondary structure from, I believe it's cricket paralysis virus, where 5' of the coding sequence, the RNA folds up into a unique structure, which tricks the host ribosome into translating it because it mimics the 5' cap. On the top left, we have something called stop-go, which is also found in a variety of viruses, most notably foot and mouse disease virus. And what will occur is that during translation, a certain amino acid sequence will occur. It's a conserved sequence that will cause the ribosome to not incorporate amino acid. So it'll end its current peptide chain, but then still continue translation. So in effect, you generate 2 different proteins. And then in the bottom left, I've not done perhaps full service to upstream open reading frames, but here's just a brief figure showing that there are a lot of different types of UORFs. So you can have non-overlapping upstream open reading frames. You can have open reading frames which overlap but are out of frame with each other. And then you can have UORFs which are in frame and can create an N-terminal extension to the traditional CDS. So we have a ton of different translational diversity. And what I want to talk about is ribosomal frameshifting. So what are the elements of a ribosomal frameshift signal, which I'm going to abbreviate as -1 PRF, programmed -1 ribosomal frameshifting? So we have 3 primary elements. We have a slippery site, which consists of a heptanucleotide sequence. We have a 5 to 9 nucleotide spacer region. So the slippery site is in the A and P side of the ribosome, and then we have a 5 to 9 nucleotide spacer, which threads through the entry channel of the ribosome. And then we have a downstream RNA secondary structure, which kind of causes the ribosome to stop. So during translation, we have our tRNAs over the A and P site. And what will happen is that while it's encountering this RNA secondary structure, it's paused. And then due to the tension that this downstream RNA secondary structure exerts on the ribosome, the tRNAs can sometimes go into the -1 frame and then continue translation in the -1 frame. And so frame shifting is found ubiquitously in viral genomes. I think most notably, now COVID coronaviruses use ribosomal frame shifting. Frame shifting in viruses was really first, I wouldn't say discovered, but really well established in HIV and Rous sarcoma virus by Tyler Jacks and Harold Varmus. And here's just a huge host of different viruses that use it, from influenza to whatever you can find on this. And what I want to highlight here in the bottom right is the first high resolution structure of actually the SARS coronavirus 2 RNA secondary structure in concert with the ribosome, which was solved by actually a good friend of mine, Pramod Bhatt, in John Atkins' lab in collaboration with Ninad Ban. So it's a really great structure. I would highly recommend you guys check that out. But what about humans? So what are the known -1 PRF signals in humans? Well, there are viral elements that exist within our genome that are left over or vestigial from previous viral integrations, and some of these sequences turn to genes or they just exist as elements. And so since this is found ubiquitously in viruses, it makes sense that in these retroviral derived elements we have frame shifting that occurs in them, though it's not necessarily sure if that's actually functional for any sort of relevant protein product, but it does exist, and they frame shift. But in terms of the first putative human -1 frameshift signal in a cellular gene, it was first discovered in 2014 that the CCR5 gene, the co-receptor for HIV, harbors a ribosomal frameshift signal. So if I'll just take your attention to panel A, we see that here HIV is a control because HIV is a really well-established frameshift signal. We see that CCR5 appears to harbor frameshift activity a bit higher than HIV. So, very, huge discovery. Frameshifting, which is this viral mechanism, is also found in humans, has broad implications for understanding this translational phenomena as a whole. And so now I kind of want to get into re-examining these results and understanding, because this is a very big discovery, is it necessarily a frameshift signal or is something else going on? And so dual luciferase reporters are really, really common for measuring any sort of translational phenomena. So on the left panel here, what what you'll have is a control vector, which you have RNA luciferase and firefly luciferase basically sandwiching either a control sequence or a recoding sequence, or a test sequence. So in essence, what you can do is you can put renilla luciferase and firefly luciferase in different frames. So let's say I have a -1 frameshift signal, and that signal is supposed to push ribosomes into the -1 frame around a rate of, say, 6%. Well, if I insert that, that RNA sequence in between, I should get renilla luciferase, and I should also get renilla luciferase and firefly luciferase fused protein. So I should have some renilla luciferase activity as my control, and firefly luciferase as kind of the dial that tells me how much recoding is going on. And here we see how this efficiency is calculated, which is simply just taking the ratio of the luciferases in the experimental vector, controlling it for our in-frame control vector. And so when we obtained these same constructs, we found some peculiar results in that the wild type sequence harbored frame shifting much higher than was previously reported, and the negative control was not so negative. It actually appeared to have a higher frame shifting efficiency, 43%. But then the HIV construct was in line with previously reported data, around 6%. So looking here at the renilla luciferase values, so renilla luciferase is our reference. We see that the CCR5 sequences appeared to have a lower reference value. So what's going on? Why is the reference messed up here? So digging deeper and looking at both the protein sequence and the mRNA sequences from these constructs, doing Western blots on both the constructs to start off with, the arrows point to the renilla luciferase, the reference. So we see that in this full length control vector and in this HIV vector, we have very clear bands for renilla luciferase, but we see no real clear bands in our two CCR5 vectors. And digging deeper, doing RTPCR on cells that have been transfected with construct, we find that the vector, if we transfected the mRNA made from the vector, we should be able to RT-PCR about a 2 kb fragment. We see that in our control vector and HIV vector, the band is very sharp and very dense. But what we see here is that in the CCR5 constructs, we see that the band is hard to see, and we see that there's this smear of lower molecular weight bands. And so just doing sequencing on these PCR fragments, we find that actually in this construct with CCR5 inserted, there's actually a splicing that's being stimulated, which is potentially creating an artifactual result. So this is just a sample of what some of the splicing constructs look like. But what's happening in essence is that renilla, the reference, is getting spliced out, and the firefly luciferase is getting spliced in frame. Now, to pause, this is really bad, because what we want is we want to see a full length renilla and firefly construct with a CCR5 construct sequence in the middle. If this splicing is going on, it's hard to interpret the results of what we're getting. So looking at bioinformatic analysis, another way to kind of double check whether or not a sequence is important is whether or not this RNA sequence is subject to purifying selection. And what we see here is that at a single nucleotide or 10 nucleotide resolution, where it's been smoothed over, the highlighted portion shows the RNA sequence, which would correspond to this frameshift signal in CCR5. We really don't see much difference in terms of the nucleotide identity. So it's really hard to say whether or not this frameshift signal is subject to extreme purifying selection compared to the rest of the mRNA. Similarly, just doing this is hard to see, I know, on Zoom, but doing an alignment of the sequences from all different mammals, we said the slippery sequence is really not well conserved either. We can see that there are substitutions in locations that would hypothetically disrupt what the motif should be and not allow frame shifting to happen. So it doesn't appear that phylogenetically there's a clear conservation of this RNA. And looking at ribosome profiling, we see that it's inconsistent with what we would see for frame shifting. So actually on the left-hand side, I've made a little figure here from TripsViz, which is a great publicly available ribosome profiling tool made by some great researchers from the University of Cork in Ireland. And this is from Pasha Barnard's group. And so PEG10 is one of those retroviral frameshift signals that I showed earlier. And so the colors represent different reading frames of the mRNA. And what we see here is that PEG10, blue is the dominant frame, And that we see here is that when we encounter this frameshift site, we see that the dominant frame becomes green, which shows that frame shifting has occurred. So we have one frame, and the ribosome profiling picks up that there's a frame shift. Now, if we look at the same sort of plot from CCR5, we really don't see an increase in reads from the alternative frame. And this is from monocytes, which are the supposed best cell line to look at CCR5 expression. So essentially, we remade the constructs in an updated dual luciferase vector, and we made our, cloned them, and then we had negative controls where we put a stop codon prior to we mutated the slippery site. We put a stop codon before the test sequence. We put a stop codon after the test sequence in the -1 frame, and then we had an empty vector. So we just use these vectors to establish a baseline level of activity. And then doing DNA transfection with these constructs in 2 cell lines and using HIV as a control. We said HIV, still has frameshift activity as we'd expect, but in both cell lines, CCR5 has recoding activity or frameshifting activity below that of our negative controls. And just to be super, super safe, actually the way this project came together is that this is a collaboration between 3 different labs. I was at the University of Cambridge, and we collaborated with the groups at University of Cork, which was John Atkins' group and Pasha Barnov's group. And then we collaborate with Jeffrey Keefe's lab back in the United States. And so these are results separately obtained from the University of Cork in Ireland, where they had actually independently been trying to figure out what's going on with the sequence. And independent cloning, independent assaying also reveals that with their one negative control, which was a slippery site mutant in 2 different cell lines, there was still background levels of frame shifting activity. And just to really double check, making sure that we're not crazy, everything's okay. Looking at frame shifting activity from these previously reported retroviruses that exist in the human genome, or these retroviral elements, I should say. We see that frame shifting activity is pretty much in line with what was previously published by different groups. So the new vector we're cloning things into, which does not have splicing issues, still reports activity well for HIV and our human controls, but still does not report activity for CCR5. And so just to be really, really safe, I made T7 transcripts from the DNA sequence, and then I transfected that RNA directly into cells. So I could then run that RNA on a gel, make sure it's a single band, and then transfect that directly into cells. And what that did was it made a really clean assay where the negative controls were absolutely at no recoding activity or frame shifting activity. They're synonymous with each other, those 2 words. But CCR5 unfortunately did not have any activity. And Western blotting of the constructs, we can see that our our in-frame controls, which are kind of our reference or experimental or our control vectors, which are forced to make the full length product 100% of the time. We see for HIV and CCR5 in these forced positive controls, we get a nice band where that firefly luciferase should be. Our wild type HIV sequence has a much smaller band because it only frame shifts about 3 to 4% in our hands. And then we don't really see anything for CCR5 or its negative controls. And again, then using in vitro translation, we kind of get the same result where the -1 frame shift band is this topmost band. We see one for HIV. Obviously, we have these forced positive controls on the left and right hand side. We don't really see much for CCR5 or its negative controls. So really the 3 takeaways I want to give from this research, which has been, essentially a lot of this has been just following up, a really, interesting result that was published previously, but by no fault of the original authors, from what we can tell, does not appear to be consistent with their original conclusions. And I guess the key 3 takeaways are always double check the system you use, have orthogonal approaches to verifying results. And this is more specific to RNA, which is important RNA structures should always be not always, but should for the most part be subject to evolutionary purifying selection. So if it's important, it should be conserved way more than say other parts of the RNA sequence. And actually, this is the first time I'm presenting this data in its whole, and hopefully this will be online maybe in a few weeks. But I just want to acknowledge all the other authors, my co-first author, Gary Loughran, Dr. Steckelberg, who's just started a group at Columbia in the RNA, the RNA subdepartment at Columbia, and then obviously a whole host of other people, most notably Andrew Firth, who was my advisor during that time, John Atkins, Jeffrey Kieft, and basically everyone else in those labs. I've shamelessly decided to promote myself twice, both on the intro slide and the acknowledgement slide. And these are just some nice pictures that we were able to obtain from a bunch of different places. And I think I'm just on time, so I'll end there and thank everyone for their time and attention.
Well, thank you so much, Yousuf. That was a great seminar, and congratulations to your paper that's coming out. That's really good.
Thank you. I think it's very rare to not have negative data, but to present kind of counterfactual data. So it's really nice that if it's something important that the journals are willing to look at it and consider it. Right. So yeah, it's hopefully will be informative to the field.
Yeah, and please post some questions in the Q&A on the Q&A tab. I was going to start with one question. So you talk about complexity to enrich the proteome. There's many different mechanisms to do that. And some of them are more scheduled and planned, but the fidelity of translation itself also leads to diversity, right? I mean, there's quite a bit of mistakes taking place when you build your polypeptide, and so you do build kind of a whole set of different proteins, a little bit different, and it could be valuable to have that kind of diversity of just having a few mistakes that makes a protein a little bit different. What's your thought of that? That maybe nature kept that in just to make the diversity a little bit more?
Definitely. I think, and actually, I don't know the exact number, but I believe it's anywhere between 1 in 10,000 to 1 in 5,000. That's like the percentage or odds that the ribosome, I think, will slip back by 1 nucleotide, but that's not a very accurate number. I certainly agree that there are instances where this frame shifting or any sort of mistakes in translational fidelity happen all the time in the genome. And it is good that it's maintained relative fidelity, but not complete fidelity, because it might be too energetically costly. But again, the thing I kind of want to harp on again is that the signals that really take advantage of this, which are mostly viruses, might be kind of taking advantage of a backdoor system, are really highly conserved. So there might be actually elements in the human genome or random sequences that might stimulate this frame shifting at a significant percentage, but the protein product is probably something that's not very stable and is quickly degraded. So again, I'm just kind of tying back to the original thought of that, highly conserved RNA sequences, which you can detect bioinformatically, I think are really the way to differentiate between this real and this maybe accidental leakage that happens through translation of the genome.
Let's see here. Anybody raising their hand or want to step in?
Yeah, I see Rahul from Wisconsin has raised their hand.
Hey, please unmute and ask your question.
I don't think we can hear him.
Can we unmute him? Martina, can you unmute Rahul?
Yes, I'm trying right now. I think, Rahul, you should be able to click it now. Perhaps you can write it in the Q&A.
Yeah. So how many years have you been at Stanford? It's just 2 or 3 years now, or how many?
Yeah, so currently I am a second-year PhD student at Stanford, where I am focusing still a bit on the RNA work that I enjoy so dearly, and also doing a lot of structural biology and protein biophysics. So a good mix of both things. I think studying RNA is a really great just primer to go into any sort of study of any macromolecule. It's just such a diverse molecule. They can do so many different things. It makes you think in many different ways. Yeah.
But the work that you're going to publish now in Nature, that started at Cambridge? That's with Andrew
So
Firth?
this work is actually, so this work is primarily done at the University of Cambridge, yeah. And obviously it's not just me, it was basically a ton of different labs that came together to kind of figure out what was going on slash, what was the issue. But yeah, I think, again, just it's important to be very careful with, yeah. Oh, weird, I cannot see Rahul Vivek in the participants list. I can see him in,
he's got
in the top here.
Yeah, I can see it too.
If he unmutes, he can talk.
If you want to ask,
maybe he's having issues.
Yeah, it seems like you're muted. So if you can unmute and ask your question. Do we have anybody else want to jump in and ask a question? Please go ahead.
Looks like Rahul's having difficulty, but if he rejoins, he can ask in the Q&A, and Yousuf, you can continue to answer questions in there as well, correct?
Sounds great.
So I don't see any more question here. So enjoy your time in Sweden now. And, you have to, when you go to bed, you have to put down the blinds because you have almost the midnight sun out there. So it's pretty light all day long there.
Yeah, I can still see out the window right now. There's still sunlight, so I'll do my best. But
Yeah.
Thank you guys for your time so much. Again, feel free to, I didn't leave my email anywhere, but send me a private message on Twitter or, any sort of, any sort of thing. So again, feel free to reach out, and thank you so much for your time. And I'm sure Mike's talk will be really amazing, and I'll let you guys intro him. Thank you.
Thanks, Yousuf. Okay. So our next rising scholar is a graduate student from University of Michigan in the program of Cellular and Molecular Biology. It's Mike McMillan. He did his undergraduate work in molecular biology at the University of Massachusetts at Amherst. And he's now working in Sami Barmada's lab and doing some very interesting work on ALS that he's going to tell us about today. And there's his title, Intersection Between RNA Methylation and TDP43 Mediated Toxicity in ALS. Welcome, Michael.
Thanks, Mats, and thanks everybody for having me. I'm excited to share some of my work today. Let me just hide something real quick here. Great. All right, yeah, so once again, thanks for having me, and great talk, Yousuf, there. Kind of a good lead in to the RNA world that I'll be talking about. And so first off, let me just give a brief background on ALS. So amyotrophic lateral sclerosis is the disease we study in the Barmada lab. And this is a neurodegenerative disease in which upper and lower motor neurons die, and this results in a patient passing away typically 3 to 5 years after diagnosis. It's an interesting disease in that around 85 to 90% of these cases are sporadic, meaning there's no familial inheritance, and then the other 15 or so percent are due to mutations in ALS causing genes. And currently there are very limited therapeutic options, whereas they only extend lifespan a couple months or so, and there is no cure. But despite this variance that we see in the presentation of disease, nearly 95% of ALS cases have TDP43 pathology. And so TDP is one of these proteins that I'll be talking about later that is involved in a lot of RNA homeostasis pathways. And then the pathology is very different than its normal kind of staining. So in this, this is a human spinal cord sample where we can see all kinds of forms of TDP pathology shown in brown. So this is what we see under normal conditions, a nice nuclear TDP stain. You can't see my laser. Right here. So this is our TDP staining in the nucleus, where it should be as a nuclear protein, and this is where it should be in a normal healthy individual. However, in ALS, we sometimes see these large cytoplasmic inclusions and these kind of spindle-like fibers that form as a result of ALS, and we don't know why these occur. And simultaneously, we can also see this nuclear depletion where we don't see any TDP within the nucleus nor in the cytoplasm. And so, this is very interesting for us because, we see all these cases of ALS with TDP43 pathology, but the underlying mechanism is still fundamentally unclear. And so when we look at what TDP does, so a normal full-length TDP protein has two RNA recognition motifs. It is first identified as a DNA binding protein, but now we know it also binds RNA as well. And then it has a glycine-rich domain in which most of the ALS-causing mutations are found, and this area is believed to be important for protein-protein interaction. So TDP has a number of roles within the cell, all kind of related to this RNA homeostasis pathways. It is involved in alternative splicing, where it acts as a splicing repressor. It is important for microRNA biogenesis, and as well important for the regulation of stress granules. And so when we kind of take all of these functions and kind of pull them into one overarching theme, that is kind of, for us, RNA stability. And so, given that we see all of this TDP pathology in ALS, we were wondering what is the stability of RNA in ALS, and does TDP, dysregulation have something to do with this? And so in collaboration with Matt's Ljungman lab and then Eva Feldman lab, we were able to look at the stability of RNA in ALS using BRUCE-seq. And so what this is enabling us to do is basically you can use bromouridine that is incorporated into nascently transcribed RNAs to give us a measurement of the rate of RNA synthesis. And then if we throw in a chase step in there, we can measure the stability over time. And so we did this in a couple of different cell types to see in ALS or TDP cell or TDP overexpression cells what stability of RNA would look like. And so in a couple of our systems here, so in iPSCs and in eye neurons, we can see a couple of things that stand out. So first, in our iPSCs, we see a reduction in stability in the ribosome and the oxidative phosphorylation groups in both TDP43 overexpressing cells and cells from an ALS patient that we reversed into iPSCs. And this is similarly the same in eye neurons, both in familial and ASLS. We see reduction in stability in these two main categories. So this was really interesting for us. We have a clear phenotype here where these transcripts related to the ribosome and the oxidative phosphorylation pathway seem to be almost regulated in ALS or by TDP43. And so we were curious as to kind of what could be the reason for this. And around the same time, a paper came out that knocked out an RNA modification methyltransferase. So it's a N6 methyladenosine transferase called METL14. And so in this paper, the authors knocked this out in mice striatal neurons. And when they did this, they found that these same two groups of RNAs that are destabilized by TDP and in ALS are also upregulated in this condition of this METL14 knockout. And so now we kind of have this kind of relationship where the stability is downgraded or destabilized in ALS, but potentially upregulated when we knock out this RNA modification methyltransferase. And so we're kind of curious, okay, so what is M6A and kind of what is its potential function in ALS and ALS pathology? So a little bit of background on M6A in case you aren't familiar with it. It's been the center of my universe for the past 4 years, but I imagine not everybody else. M6A is the most abundant RNA modification that we know of to date, and it's been implicated in a number of diseases, and it's really kind of emerged as this new hot topic in RNA biology. So there has been cases of upregulation of M6A in cancer. It's been known to be important for development. There have been mutations in M6A related proteins that can lead to obesity. But I'm particularly interested in the role of M6A in neurodegeneration. So given M6A is this RNA modification that throws onto this 6th position of adenosine, it has a few important roles in RNA homeostasis, splicing, translation, and then also really involved with export and decay. Specifically, RNA decay and degradation being the forefront impact of M6A on RNA. And so when an RNA becomes methylated, or hypermethylated or hypomethylated, this can change the fate of that RNA, and that is really the importance of M6A in the cell. So when we look at M6A, it's deposited at these DRAC motifs. And so this is a pretty common motif throughout the RNA transcript. You'll find a bunch of these. However, only like 1 or 2 or maybe 3 are typically actually methylated. And we still don't know the reason why, but we were able to determine where this sequence is. So in this paper by Kate Meyer, what they're able to do is use MeRIP-seq. So this is an M6A IP, and then followed by some RNA sequencing to determine the location of these M6A sites. And so using HEC293 T cells and mice brain, they were able to see that we have an enrichment in M6A in the coding sequence and in the beginning of the 3' UTR. And so this dashed line here, this is actually the stop codon. And so kind of makes sense that there would be an enrichment in M6A at the end if this has future roles in splicing, polyadenylation, or RNA degradation. And then when we look at where M6A is in terms of tissue type, we see that M6A is the most enriched in the brain, which is really interesting for us if we're interested in seeing how does M6A impact individuals with ALS. It's good to see that there is an abundance of M6A that we can work with. And then so TDP43, right, it has all these roles in RNA biology. It has been known to bind hundreds and thousands of RNA substrates. And we wanted to see, what percentage of those substrates could be methylated to give us an understanding of, is there some sort of relationship between TDP43 and M6A? And so when we look at a few different published iClip data sets, we can find that in most of them, and up here, this other one, there's like 30 to 50% of these RNA substrates that were identified have at least one M6A peak. Some maybe have more. And so this is kind of interesting, and we're curious to see, does this methylation decide the stability of those peaks, of those gene targets? And then this is kind of the basis for my thesis work here. Does TDP bind and destabilize these methylated RNAs? So with this understanding that there's this large data set of TDP being able to bind RNAs, we wanted to choose one specifically and one that we know it binds pretty well. And TDP binds itself on its 3' UTR, and it's happens specifically at these 2 regions that have been shown to be sufficient to elicit TDP binding. So I was curious, what, if any, methylation sites are around these areas. And so we overlay some known M6A data. We can see that there are 2 methylation marks shown by these red pins, specifically within these TDP binding regions. And so this was really interesting for me, because now, if you think about it, what happens if we get rid of these M6A sites? Will we then be able to prevent M6A binding if TDP does bind to methylated RNAs? And so that's exactly what I did. So I tested out in HEC293 cells a 3' record that we have for the TAR-DBP gene. So I transfected these cells with TDP tagged with a halo tag on the end, plus this wild type reporter. Did a pull down and performed qPCR to look for the abundance of TAR-DBP here. And we can see under our wild type conditions that we have this nice enrichment of TDP or TAR-DBP mRNA, which is expected. However, when we throw in this mutant, so when I performed site-directed mutagenesis to change this A to a G, in a sense to eliminate this methylation site, we can see that the mutant is basically unable, it's not barely above background compared to the wild type version. So this methylation site is potentially really important for eliciting TDP43 binding. And so kind of future directions from this area and some data that I'm waiting to get back from. Recently, I performed DART-seq pull down. So DART-seq is a deamination adjacent to RNA targets. And this was another method pioneered by Kate Meyer, where you transfect in a construct which has a YTH domain. So this is an M6A binding motif. and then this ApoBec1 protein tag on top of it. And what this does is induce C to U transitions. And so transfecting these into cells, you can then monitor or identify M6A sites independent of antibodies, which is going to be a really big step there because of the specificity issues that a lot of M6A antibodies have. And so I applied this to our Halo TDP cells in a sense to, one, identify TDP RNA substrates, and then on top of that, their M6A signatures. So being able to identify to what degree of methylation they have and then where those methylation sites are. And additionally, also performing an EMSA with that TDP binding region, plus or minus a methylation mark there, and it's to see if that is sufficient in generating more TDP binding. And at a similar approach, we're going to be collaborating with Andreas and Niels Walter's lab, where we'll be doing this in cells. So we'll basically be in vitro transcribing an RNA probe, and then monitoring the ability of TDP to bind to this probe, plus or minus a methylation mark on that as well. So that's really exciting there. And so that is basically looking to see if TDP is able to bind our methylated RNA transcripts. And so taking it a step further now, right? So we have this understanding that potentially M6A is involved in generating this TDP binding to RNA substrates. But what does that look like for ALS as a whole, right? How important is that to ALS pathology? So I then wanted to take an approach to basically determine if increased methylation could be destabilizing some RNAs, and this is why we have more neuronal cell death via TDP43. And so to do so, I used our longitudinal fluorescence microscopy platform, where I transfected rat primary cortical neurons with metal-3 and metal-14, or any other kind of plasmid in this sense, and then we can monitor their survival over time. And in doing so, we're able to track neurons from the beginning all the way through their life cycle and identify their time of death, both as a population and on a single cell basis. So when we do this, I was interested in seeing again, right, if we can increase methylation, will that lead to more cell death because of this involvement of M6A and RNA stability? So under normal conditions, if we transfect in our TDP43, that leads to more cell death, and this is a cumulative risk of death plot. So the higher the y-axis value is, the more cell death there is. And we can see compared to our controls, we have a significant enrichment of cell death here with TDP43 overexpression, which is a very good sign for our experience here. And then when we throw on metal-3 and metal-14, so these are the primary components of the methyltransferase complex for M6A, really metal-3 being the enzymatically active methyltransferase, we see that while there is not really much of an effect down in our controls, we see this large enrichment, a significant enrichment in toxicity associated in the TDP43 condition. So on the background of TDP43, enhanced methylation is worse for the cells. They don't tolerate it, and can potentially be an identification that hypermethylation may be significant results for neurodegeneration. So going further, right, I then wanted to see, okay, so now I have this evidence that methylation is worse. What would happen if I can try and prevent that, right? So to do this, I performed a CRISPR screen, candidate-based CRISPR screen in primary neurons, so the same rat primary neurons we've used in our transfections, and then knocked out some of the main components of the M6A pathway. So in the left here in green, these are some of the writers. So you're familiar with metal-3, metal-14, right? These are the enzymatically active writers, part of this complex. We have some demethylases, some erasers, FTO-BH5, and then in yellow, both nuclear and cytoplasmic readers. So these are M6A RNA binding proteins that once they bind to the RNA, it will take it in a number of different ways based on the function of that protein. So for instance, YTHDF2, this is a RNA binding protein involved in RNA degradation. And so I just took some of these main components and design guides against them and to figure out if knocking any of these out would be able to rescue some of the TDP43 toxicity that we see in ALS. And unfortunately, most of them, made things worse. We really saw a lot of toxicity with the majority of them, except for one in particular, and this is YTHDF2. So when we overexpress our TDP43, we see more cell death. But then when we introduce this YTHDF2 knockout on top of it, we see a rescue of TDP43 toxicity, and this is at near physiological levels. And then in our controls, again, we don't see a significant result here. We don't really see much of a change. So this seems to be a TDP43 dependent result, much like what we saw with the overexpression of M6A via metal-3 and metal-14. So this was really great to find, one piece of the puzzle there that maybe could help lead us to, what methylation and TDP43 are doing. And so this has been so far in HEX cells and in rat primary neurons. We wanted to then take it a step further into a human-based model, right? A more physiologically relevant model. And so we used our iPSCs, differentiated them into eye neurons with some help in the lab by Michael Beckier. And so this is just under normal conditions with a scrambled shRNA. We can see our C9 ORF72. So this is the most common familial version of ALS. Under these conditions that, it is more toxic. We have more cell death. However, when we introduce the YTHDF2 knockout or knockdown on top of this, we see a really incredible reduction in toxicity in the sense that it brings it below our background control levels. And so this is really, really great. And this is preliminary evidence so far. This has only been one experiment. We have a couple of these coming up. Michael's been doing a great job. But this is, potentially the first evidence that YTHDF2 could be a therapeutic target for preventing some of this toxicity that we see in ALS. And then, this has been great data so far. And what does YTHDF2 look like in ALS or in spinal cord and cortex? We don't really know. There hasn't been a whole lot of staining for that. So that's exactly what we did next. And looking in control in patient cortex and spinal cord, we actually saw something really amazing. So first off, in our controls, we're looking at YTHDF2 stain. So it's in the brown staining, you'll be able to see that. In the cortex, it seems kind of faint in the control, and it seems, like a little bit darker in the ALS patients. And then same thing we see in the spinal cord. And really, when we look at some of these patients in the ALS spinal cord, we see kind of this punctate formation and this really different phenotype of YTH. Typically, it's this kind of nice cytoplasmic kind of perinuclear staining. However, in our ALS and particularly in our spinal cord, we see this really different staining, and we're really trying to validate this with some additional samples. But so far, this is kind of some really striking results that there is potentially, upregulation of YTHDF2 in ALS, and potentially a target for a future therapeutic intervention. All right, so now we kind of have an idea that we have evidence that hypermethylation via metal-3, metal-14 overexpression could be is more toxic to our cells and enhances with TDP43. We have evidence that TDP43 binds or needs methylation to bind to RNA substrates, and then YTH being upregulated in ALS as a M6A binding protein. Is there more methylation around? Is methylation being upregulated in ALS, and could that be a reason we see some of this pathology? So we wanted to test this out using an epitranscriptomic array. And so what we did was we had some control and ALS spinal cord. We extracted the RNA from this and sent this out to perform this array in which we're able to kind of differentiate both the M6A and the non-M6A RNA transcripts to give us an idea of the fold enrichment of M6A across different transcripts. And so when we look at some of the results from that, first we see that in these hierarchical clustering graphs that our control and our ALS patients tend to cluster together, right? So there is evidence that there is some sort of different profile going on here. And then when we look at a principal component analysis plot here, we can see two distinct populations. So this gives us an indication that there is really strong differences in these methylation profiles. And looking at the RNA transcripts themselves, we see this really striking result of this hypermethylation that's occurring. So these are some volcano plots that are plotting the methylation of all these RNA transcripts from the array, and anything above this line is significant, and anything to the right of this line is significant. So these are our hypermethylated transcripts. And so in our mRNAs and long non-coding RNAs, we really see this abundance of about 30% of hypermethylation going on here. And this is, the first evidence that there is hypermethylation in ALS. Potentially, there's some dysregulation of the M6A related pathway going on. And then when we throw that on top of some of our previous data, looking at the stability of RNA from the BRUSeq, you're looking at our familial ALS that was been destabilized, and then with TDP43, we have 26 genes in common, and kind of maybe unsurprisingly, these relate to ribosomal genes and mitochondrial related genes. So again, bringing back the importance of those ribosomal and oxidative phosphorylating transcripts from those initial stability results. And so this kind of leads me into my model of what I think is going on here. So if you imagine under normal conditions, your genes are transcribed, and there are a couple different fates for the RNA. So under normal conditions, right, you can have unmodified transcripts being spliced and then translated, whereas the modified transcripts are led to degradation. And so kind of what my research points to is that in ALS, right, we may still have the same amount of ideas here of splicing and translating, but there is hypermethylation occurring, and that could potentially bring TDP on to target some of these RNA substrates, and then the overall number of these modified transcripts is different, right? So say it goes from 90-10 to 40-60. So now we have a larger pool of RNA substrates that are modified, which may not need to be modified or were not previously modified in the past, leading to kind of this global dysregulation in RNA stability that we're seeing via our RNA stability results there. So that's kind of where things are at right now, and I have some hopefully some of that data coming back with the DART-seq and the MSAs can help validate that claim. But this has been really interesting work so far, kind of diving into this M6A pathway and being able to see that there is some evidence that this could be a really good therapeutic avenue for targeting to treat ALS. And so with that, I'd like to thank my lab for letting me talk about M6A all the time, even though they probably think they don't want to hear it anymore. They're going to hear it a lot more. And then, my collaborators, the Feldman lab and the upcoming collaborators with the Niels Walters lab there. And yeah, I'll take any questions. Thanks.
Well, thank you, Mike. Great talk there. So if you have questions, post them in Q&A or unmute. We're not that many online here, so we can unmute and ask questions. I guess I could start with one question. So, I mean, you showed, your previous data from the lab that, in ALS, the ribosomal transcripts and mitochondrial transcript seems to be less stable than in normal situations. And I was just wondering, when you do, for instance, the knockdown of the UTH, Do you know if that then protects the turnover of these groups of transcripts?
Yeah, that's a good question. I haven't looked at that. I don't know. I haven't really looked at that. So it's kind of interesting because in terms of methylation, the smaller transcripts are methylated less, and a lot of those smaller transcripts are ribosomal transcripts. So potentially knocking out YTH could reduce those, but it's also possible that those are not methylated under normal conditions, but in ALS they may be. So that is something we can look at definitely too and see what's going on there.
We got one question here from Yousuf, who talked before here. He said, hi, Mike, thanks for your talk. So if I understand correctly, the idea here is that TDP-43 binds methylated RNAs, but then what? Do you think it's leading to degradation of the mRNAs? Does it have a domain that could have nuclease properties? Or do you think it could form a complex with something that could eat the mRNA?
Yeah, and that's a good question. And so I think, TDP itself doesn't have any nuclease activity, but I think it acts or could act in a complex of things. And so what's interesting for me and what I'm looking at right now is this interaction between TDP and YTH DF2. When YTH DF2 binds RNAs, it takes them to P bodies to be degraded. And so if you can imagine YTH being primarily nuclear, or TDP being primarily nuclear, YTH being primarily cytoplasmic, maybe there's some sort of handoff mechanism there where it gives an RNA substrate to YTH to be degraded. Or when TDP shuttles out of the nucleus, maybe it takes it to some of these RNA degradation areas. So I don't know what the answer to that is yet. We're working on it, but I definitely think it acts in a complex or in coordination with some other proteins to kind of degrade that RNA.
We have a question from Peter Todd. Do you want to ask it, Peter?
Sure, how you doing, Mike? So your C9orf72 data is quite compelling. What do you think that is? Do you think that's a TDP-43 effect, or do you think it's independent of that? And do you have any data to support that, your statement about it?
Yeah, so this is like really preliminary data. Michael has been working a lot, but he's kind of hard to work with. So I don't know if it's TDP-43 dependent or independent or not. I don't know what there has been some evidence out there that I've seen that maybe there is a different kind of methylation profile in C9 than other forms of ALS, but it's kind of too early for me to say anything on that.
Okay, we have one, I think this will be our last question from Yin Lee. Nice talk, Mike. The DF2 increase you observed was in cortex from C9 ALS patients or from SALS with TDP pathology?
Yeah, so this is from, I believe it's sporadic ALS patients. So we're currently in the process of staining some of those similar samples and trying to do a side-by-side staining with TDP and YTH-DF2 using immunofluorescence in these samples to see if we can, see TDP pathology and YTH pathology, I guess you could call it, or differential expression in these same samples. So that's something we're currently working on now too.
Okay, I don't see any more questions. Well, thank both to Yousuf and to Mike for great presentations. And this was the last RNA Innovation Seminar for the season. We start up again in September, but we do have one RNA Collaborative Seminar series on June 30th, I believe. So stay tuned for more information on that. So with that, thank you so much for everybody listening and for our two rising scholars. Good luck with your work.